Alright. See if I can't show you guys some stuff. Get the thing hooked up there. There we go. Let's see what we got. Hopefully the music's not too loud for anybody. I like it. Zoom the heck in there. So it looks like we've got some yeast cells in here on my slide. What I did was I added some methyl blue to the yeast starter solution that I put on the slide. This is a humidicytometer. I believe is how you say it. Basically cell counting slide. It's basically got a big old grid etched into it, and you just count. You just one by one count all these guys. The methyl blue, methylene blue. Excuse me. What it does is sorry. There we go. Alright. Let's see here. So anyway, so when you have methyl blue, which is, I'll show you that right here. Let's see if we can see that. This is just what I got here. It's just methylene blue there. Basically what that does is it dyes the cells. That's what I learned when I was up at the beer boot camp up over at Santa Rosa. Dr. Chris White from White Labs taught the class. We had seen him, and he asked all these questions. Why you guys aren't counting yeast cells? Why aren't you counting yeast cells? And nobody has a microscope. Well, you can get a pretty cheap one, and I got this one for pretty cheap. I forget what I paid for it. But it wasn't over $100, I can guarantee that. Came with a cell phone mount, which is what's holding my cell phone right now. So, which is good. And so I can have a free hand. I can count cells. I can take pictures and count them later, which is also good. It's pretty awesome. It's the first time I'm using this all together here. But, yeah, so far it's been pretty good. I've just been counting cells this evening, figuring out, you know, what we've got going on inside my yeast. See if they're healthy. The whole reason behind that is to, you kind of want to take a notice. I posted a little bit of this sample earlier, and it looks to be swirling pretty good. I actually have it right here. Let's see if you can get this in a shot here. 2-liter starter. And I don't know if you can see that nice, creamy, thick yeast cake on the bottom. It looks pretty good. So if it's already settling out while it's been sitting here next to me, I've been pulling from it, testing samples, things like that. So it's been pretty good so far. I've just been counting cells, averaging it out. So this starter should be pretty much good to go here in about a day. I think I'll be ready to pitch it. That IPA that I keep meaning to make. I was initially just going to make 5 gallons and just boom, drop it in, but I think since my porter's done, that's going to free up some room in the fermenters and my kegs. So I'm just going to bottle all that and then just distribute it out to friends and family or whatever if they want. I don't know. Give it away. Because it's more than enough beer that I can drink. My porter came out pretty good. It's my first porter I've made. I think it tastes pretty good comparative to other stuff. But yeah, let's get back. Let's see some more yeast cells. And then we can. Let me adjust the camera here. There we go. That's one thing. This thing kind of doesn't really work all that well on the cell phone. Yeah, but let's see. Oh, cool. Looks like I can zoom in, which is awesome on this live video. So wondering if I could do that. So you've got the grid square right there. The blue, what this methyl blue does is the yeast takes it in and then it metabolizes it. And the healthy cells are supposedly in white. The blue cells are dead or they're very young. It's kind of like the catch-22, and if I see this and I see this, it was nice and healthy and active yeast in my starter, and I see these are light blue, according to what I learned in the class, these are more than likely really young, just separated off. But the thing is that way too many, I don't have enough of the plain white cells to judge off of as to where they came from. So I don't know right now if that, let me move some more. See if I can find one that's a lot darker. At this point, we'll see here. See what we can find. Yeah, it's a little bit darker over there. That guy there. Off the track there. Go down. That's the end of that. Some nice cells over here. So I don't know if... I don't think these are just fresh buds. It's air pocket there. My slide. Ruin that frame. I don't know. Some nice white ones there, but see, you've got all these blue cells, and it kind of bugs me a little bit. I don't know if changing the shade is really going to fix that. Oops. Oh, I'm way off the rails now. Found my way back. Nice little pocket of cells right there, huh? Too bad it's not in the window to count. We'll find it, we'll find it, we'll find it. There we go. See? Some nice ones in there. See if I can't change the... I'm trying to adjust it to get the best look at it, and it's really cool. You can almost see the organellas in there too, which is really nice, even though this is really a cheap microscope. Let me see if I can change the backlighting there. See if we can't adjust anything. Oh, we're out of focus. See what we can do. Yep. Oh. Good. Nice contrast there. I think maybe I'm not getting a full count now that I'm playing with this a little more. So it looks like, I don't think you can really see it, but right there in the center there's one that's kind of like a paler blue, not like those white ones in the top left corner of this grid square. You know, it's 1, 2, 3, 4. It's a 4x4 square right in the center of the viewfinder. Pretty much right in the center. Let's do left to right on the top. So it's third over from left to right, and then one down. Got that cell in the middle, the black spot in it. I believe that one's a good one, even though that one is showing up earlier is kind of blue tinted. I think that one's still good. Oops. Yeah, I think that's what it is. I think a lot of these are newer cells. That's what I think. I'll have to check it in a day or so. Because they're supposed to be opaque. I wish I could catch one budding. It'd be pretty cool. I wish I had a stronger microscope. I'd be able to see a little better. It'd be a little bit easier to count, but you know, if without counting, I guess it's pretty much like makes it look a little bit better as far as like what's going on. There we go. That looks a lot better now. I like that one. Frame looks pretty good, I'd say. I don't know. So left to right, top to bottom, kind of a snake pattern. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 26. 26. Let's see if we can't find one other one in there. In that frame, it looks like it's pretty low, I think. Oh. Oop, there we go. It's a little backwards in trying to move this. It's good though. Wish I had a lot more cells. I think it's maybe just because they're so small, they may be not mature, and so their metabolism's slower. It's really hard to say for me right now without the experience that I have. There's a lack thereof, but they're not as dark blue. They are lighter blue, I would say, compared to what I was shown. I'll have to figure it out. Safe would be not to bet on them. I've almost got like a half viable because it wouldn't be dead all the way if they were on the back end. It's kind of an older colony. I may have to wait until this grows up or maybe get something else. I don't know, but it's producing pretty well at least. At least I'll maybe only get one out of it. We'll see here. Well, thanks for stopping by. If you did, I'll see you. Keep counting.